the next talk, and I request all the attendees to post the questions for the speaker so that they can be properly addressed. So now moving from the basic about the digital pathology and the image biobanking. The next session is about the advanced techniques in digital and computational pathology. And for that, I would like to invite Professor Lee, from the Seoul National University Hospital, South Korea, and she is going to talk about the advanced techniques in the digital and computational pathology. Over to Dr. Lee.
I am Kyoungbun Lee of Korea, pathologist of Seoul National University Hospital. It's my honor to take a last topic, and congratulations on the first ASDP seminar. I have nothing to disclose. I will talk about the synthetic or artificial histopathology images in the era of artificial intelligence. Traditional histopathology is based on 2D images using tissue slice. 2D image has a limitation in evaluating the convoluted structure, the complex distributed object, and the rare object. 3D pathology images are the solutions for this limitation. Is there a way to scan the tissue without tissue destruction? Two critical technology in this field are how to capture the inner content of tissue and how to visualize the captured signals in order to make the human understand. There are two ways to make 3D pathology images. The first one is staking serial section of tissues, which are tissue destructive, time consuming, and laborious. Knife edge scanning and micro-optical section tomographies are the examples of automated serial sectioning and rendering method. These methods are tried in research field. The second one is non-destructive 3D microscopy, confocal microscopy, multiphoton microscopy, and light sheet microscopies are the examples of this method. Confocal and multiphoton microscopies are point scanning method. Light sheet and open-top light sheet microscopies are scanning the 2D surface of tissues. These methods are being optimized for the clinical specimens by increasing the scanning depth and image quality. Knife edge scanning microscope and the micro-optical sectioning tomography are the automated scanning simultaneously with sectioning. Staining of whole tissue is prior to sectioning. The upper image is a mouse brain section stained by laser staining and reconstruction images. The lower picture shows axon interaction with modified Golgi and Cox in 3D reconstruction. There was a trial to make 3D pathology using 18 whole slide images. These are 3D tissue blocks using about 100 sections, gapping 25 to 35 micrometers. This virtual tissue block can make virtual sectioning with arbitrary axes and show the interaction and networking of different tissue components using various staining. However, it can be used for research only, not diagnostics. Confocal laser scanning microscopy and multiphoton microscopy can tissue without sectioning. Half 1 millimeter thick tissue was stained by fluorescent dye with or without antibody and was cleared to penetrate light. It can cover 800 micrometer thick tissue and make 3D images. The upper picture is fluorescent color, and the lower picture is a pseudo-color conversion of fluorescent intensity to RGB density. Light sheet microscopy is a non-destructive tissue scanning method. It scans the tissue surface prepared with clearing and nuclear staining. The strength of this technology is the fast scanning speed in shallow depths. The concept of this method is similar to pathological practice using representative sections after trimming of a paraffin block. Open-top light sheet microscopy is a modified technology to increase focus depth and decrease signal reflection from the surface irregularity. This method can use a color conversion based on nuclear staining, and this method covered about 1 millimeters from surface. Mid-range light picture shows the images of a prostate of needle biopsy, and the author report the change of Gleason score between the original glass slide and after using the 3D images. The main core of non-destructive tissue scanning method is tissue clearing. Clearing chemicals should not change the biochemical property of own tissues. This paper reported immunohistochemistry result before and after tissue clearing to check the influence of a clearing solution or method. Some markers, the gray and red markers, show the different result. It seems that more research is still needed to implement it in practice. These listed issues for non-destructive tissue images. In tissue clearing and labeling, time, cost, image reproducibility, and biosafety are important considerations. In the imaging system, resolution, contrast, speed, the covering area or depth of tissues are essential considerations. In data handling of 3D images. 3D images contain highly repetitive microscale features such as cells, nuclei, gland, stroma, and vessels. The scale reduction is needed using automatic quantification using an AI algorithm and based on the biology meaning. 3D rendering and pseudo-color are the main algorithms for virtual sectioning and for pathologist interpretation. Some free algorithms and commercial software are introduced. Recent AI algorithms using convolutional neural network and generative AI are used for this field. 3D or large range of spatial data of 2D pathology can be used in co-registration analysis of multi-scale medical image data. Aligning different staining sections was a challenging task in a medical image AI challenge. It means that this topic is attractive to the medical field. Overlay of immunohistochemistry on 18E or vice versa is commonly needed in pathology practice. Analysis of a multiplex image is based on the individual tissue or cell types. To do this, multiplex images should be overlaid on 18 images. AI is used to register the different stained images. Another approach to doing this is the direct classification of cell type. However, the classical supervised learning method requires many cell labels, which is laborious and time-consuming. Self-supervised learning was based on the small reference labeled data and the algorithm of similarity calculation and achieved the performance of a full supervised learning algorithm. Another example using multiple stained serial slide is research that automated 18 segmentation into tumor, stroma, and lymphocyte and quantification of specific immune markers predicted neoadjuvant chemo response in pretreated breast core tissue. Blue curve is the prediction by Impress author's model, and the red curve is pathologist prediction in HER2-positive breast cancer and triple-negative breast cancer. It used the spatial information of multiple 2D images based on AI. Virtual staining is to covering non-stained tissue images to familiar conventional staining images. It is the key skill to skip the staining process and to visualize in non-linear or ex vivo microscope. This paper tried a generative adversarial network algorithms to make 18 images from label-free autofluorescence images. first and the second gray color images are baseline images under the filter without dye, and the third column is a network output, and the fourth column is real staining pictures. Generative adversarial network is famous due to ChatGPT and is the method of learning representative data and generating new synthetic data such as text-to-image synthesis or super resolution images. It can be applied in pathology data to color normalization and make a virtual staining, such as making simple HME and predicting immunohistochemistry from the HME slide. Ink removal or image enhancement to high-resolution images from low-resolution images or unfocused images is another field to be applied again. Extract multiple pictures to generate synthetic learning data can be used to transfer the image features of untrained data, such as some features of trained data to the untrained another images of different laboratories. Segmentation of nuclei is an example of data augmentation using synthetic data from small labeled data. Data augmentation by synthetic data is one of the alternative solutions for the shortage of labeled data accompanied with weekly or supervised learning. The last technique is augmented reality microscope. This picture is the AR microscope by Google. The tumor is displayed in the eyepiece in real time during the slide navigation. Several tissue analyzing values or marigold tumors can be visualized in this method. It does not need tissue scanning, but need a high-speed inference computing. It can be used for direct collection of a field of view and analyze of the field of view. I'd like to close by summarizing some thoughts on recent AI and histopathology. Histopathologic examination is the standard for the diagnosis of disease. H&E stained slide or images are very classical method, but I think it is not the gold standard tools for getting histologic information. Non-destructive slide-free unlabeled images can be the alternative new platforms. To do this, extensive comparative validation study on diagnostic quality and cost effectiveness will be needed for the implementation of new technologies in the practical field. Deep learning and AI technologies are opening the era of visual informatics in histopathology. And generative AI is a big way to solve the current problems in machine learning in histopathology images, but the traditional chemically or biologically based histotechniques have the explainability or logics of result. The explainability of AI model is an essential factor to earn the same status of classical histotechnics in the real world. Thank you very much for your attention.
Thanks, Askari, for your wonderful lectures.